Hi everyone, welcome back to the channel. I'm the GCSE Science teacher. In today's video, we're going to revise all the key content for GCSE Biology. And if you do enjoy this video, let me know by giving it a big thumbs up, share it with someone else so they don't miss out on key revision, and please subscribe to this wonderful community. Thank you so much for your support. Let's get started. The first topic is homeostasis. We're going to talk about the nervous system. So the nervous system is made up of the central nervous system and the peripheral nervous system. So you have your brain and spinal cord in the center, and branching out from there, we have our three different neurons: motor, relay, and sensory neurons, and also the effectors and receptors. Remember, a gap between neurons is called a synapse, and neurotransmitters are chemicals that actually diffuse across that synapse. But neurons use electrical impulses to pass on and coordinate a message. They do this through a sequence of events called the reflex arc, and this is a really fast automatic response that does not actually involve the conscious part of the brain. So you need to know the following steps, and you should be also able to identify these steps in diagrams. So it first starts off with a stimulus or a change to the environment, and this is often something that could threaten our existence or health. This information is then received from receptors on, say, different sense organs like the skin, the eyes, the tongue. We also have what we call sensory neurons that receive this information from the receptors and pass the information on to relay neurons to then pass it on to motor neurons that essentially trigger effectors. These could be glands or muscles that cause a response. So if it's glands, it will produce a hormone, or if it's muscles, they will contract. Now remember, there's three different neurons in this process: the relay, the motor, and the sensory neuron, but they all have very similar structures. On one side where the information is received, we have our dendrites. These are branches that receive that information from the previous neuron. The cell body, or the soma, contains a nucleus. Remember, this is a cell, so it will have a nucleus, therefore it's eukaryotic. It also contains cell membranes and an axon, which is a very long structure. So one of the ways the neuron is specialized is that they're very long and they have lots of branches. And this axon is covered in an insulating material called myelin sheaths or Schwann cells. And in between the Schwann cells is something called a node of Ranvier. And this essentially helps ions to pass through and increase the speed at which this occurs to transfer that electrical impulse. And at the very end, we have an axon terminal, and this is where we then find the synapse, the gap between two neurons. Now, there is a required practical for part of this topic, and it is all to do with reaction time. So how quickly someone can actually respond to a given stimulus. An example of a negative feedback loop can be found in the case of glucoregulation. Essentially, this is our body's way of maintaining optimal levels of blood glucose concentration. And this is done when we eat or when we don't eat. And this is something that can happen all the time. And if it doesn't work, it can lead to different diseases. And you do need to know about those different things here. So the first thing to know is the endocrine system is made up of a range of different organs, which are called glands, because they actually produce and release hormones. These are chemical messengers that have a long-lasting effect. They are slow releasing into the blood, and it takes time for them to reach a target tissue to cause a response. So it's much slower than the nervous system, but it is longer lasting. Now, target tissues sometimes are other glands, and therefore we often call the glands that act on other glands the master gland, such as the pituitary gland, because it is found in the brain and it releases hormones to do with female fertility like LH and FSH that can actually act on other glands such as the ovaries to release estrogen. Now, you do need to know the location of these glands in the human body and also the hormones they produce and what they're responsible for. So if you are a triple scientist, however, you'll need to know thyroid hormone, thyroxine, and also the adrenal glands producing adrenaline in a bit of detail. So thyroxine is produced by the thyroid gland in the throat, and essentially what it does is stimulates the metabolic rate. It controls the speed at which oxygen and food products react to release their energy in the body, and this is what we call metabolism, the sum of all chemical reactions in the body. Now, the key to this, if you're a triple scientist, when you talk about thyroxine, is to know that low thyroxine levels in the bloodstream actually stimulate the hypothalamus in the brain to release something called TRH. And this causes the pituitary gland, which is another part of the brain, to release something called TSH. And therefore, the thyroid releases more thyroxine. And this will cause the blood levels of thyroxine to kind of return to normal. However, if there are normal levels of thyroxine in the blood, this will inhibit TRH release from the hypothalamus and the production of the pituitary gland. So normal levels are maintained. Now, this again is an example of a negative feedback loop that you should be expected to know. Likely, also, you do need to expect it to know is about adrenaline and the impact it has. So if you're a triple scientist, you need to know that adrenaline is produced by the adrenal glands, which are found on the kidneys in a time of fear or stress. And it essentially targets vital organs such as the heart to increase the heart rate, to boost the delivery of oxygen and glucose to those working muscles, but also the brain as well. And this is to kind of get it ready for that fight or flight response. However, it's really important to know that adrenaline is not controlled by negative feedback. This is kind of an innate response. So you need to know as well that adrenaline will also not only increase the heart rate, but also the breathing rate to maintain that amount of oxygen getting to those different cells to increase the level of respiration. So glycogen, a store of glucose, is actually broken down into glucose and helps with respiration being more efficient. So we have more energy to fight or flight as well. Now, if you're just a high combined or you're a foundation student and a triple student, anybody should know this, that the pancreas is another gland that's found in the abdomen and it releases insulin. However, if you're a higher tier student, you also need to know it releases a hormone called glucagon. And this is produced when the glucose is gone. There's low levels of glucose in the bloodstream. And you need to know in those cases, glycogen, that store of glucose in liver muscle cells or just cells in general, are broken down into glucose and increases that level of glucose in the bloodstream. However, insulin is released from the pancreas essentially when there is too much glucose in the blood, and it can be stored away in the cells as glycogen. It's important to know that glycogen is insoluble and therefore it's a great storage mechanism. Remember, glucose can also be stored as starch, but that is in plants, so don't get confused with that one. It's also worth mentioning at this point that you must spell glucose, glycogen, and glucagon correctly, so definitely make flashcards on those. Now, if someone's pancreas doesn't work and they don't produce enough insulin, they will have a condition called type 1 diabetes, and therefore they'll need to inject themselves with insulin. However, someone can actually develop type 2 diabetes, and this is because the pancreas is producing a load of insulin, but our body cells do not respond to it anymore, and that means that glucose levels stay high in the blood for a long time, and that's a really key indicator for diabetes in general. So someone with type 2 diabetes won't store that glucose as glycogen, and they'll need to go on a carbohydrate controlled diet and increase the activity level. Menstrual cycle is a sequence of events that lasts around 28 days, and this is a continuous process that occurs for the majority of the female's life. Now, this is a process that starts off day 1 to 7 during menstruation, where a period occurs or blood is released from the vagina, and this is because of a breakdown of the uterus lining as there is no pregnancy. And after this time, the uterus lining starts to build back up thanks to the presence of a hormone called estrogen released from the ovaries, and the uterus lining is maintained again by a hormone called progesterone. Now, at this point, an egg cell will start to mature, and this is thanks to a hormone called FSH, which is released from the pituitary gland. This is follicle stimulating hormone. And as the egg cell matures, the effects of estrogen actually inhibit FSH, and this is to prevent more than one egg from maturing. And this is something higher tier students should be aware of. Now, at this point, around day 14, there is a surge in LH or luteinizing hormone, and this is again released from the pituitary gland, and this causes what we know as ovulation. So an egg cell is released into the fallopian tubes, and as a result, a sperm cell can join it and meet to cause a fertilization event. However, if there is no fertilization event through sexual intercourse, essentially what happens is the uterus lining will break down and a period will occur or menstruation starts again. Now, if you are a high tier student, you need to be able to see the interactions between these hormones. And if you're a foundation student, you should be able to just identify those different hormones and their functions. Now, there are some hormonal contraceptive methods or methods to prevent pregnancy, and they use estrogen and progesterone in the form of the pill. And this is to prevent the effects of FSH and to stop the egg cell from maturing in the first place so that sexual intercourse will not lead to a pregnancy, as there are no mature egg cells. However, there are some other contraceptive methods, although hormonal contraception is around 99% effectively used if used appropriately, and its efficacy is high. However, there are some side effects as with any kind of medication. In terms of non-hormonal contraceptions, these are things like barrier methods, such as condoms that physically prevent the sperm and egg cell meeting. We also have IUDs or intrauterine devices, which physically stop the implantation of the embryo, and they can also sometimes release hormones as well, so they can be non-hormonal and hormonal. We also have things like spermicidal agents that kill any sperm, and also abstinence, which is just no sexual intercourse as well. And then lastly, there's also the implant, which can release hormones similarly to the pill that releases estrogen and progesterone. Now, if you're a triple scientist, you'll need to know about a method called IVF, or in vitro fertilization. And this essentially is when people who can't have a child naturally will actually use medical interventions. So what they will do is they'll take some of the egg cells from a mother, and this is after she is injected with fertility hormones such as FSH to increase the number of egg cells that mature so that there are more eggs to use to ensure that fertilization can occur as accurately as possible and successfully as possible. We also have the sperm cell that is removed from the male as well, and they are combined together in a petri dish, and they are allowed to develop over time. and embryos will be implanted. Several embryos will be taken and implanted into the mother. This can be a surrogate, or it could be the mother that's the genetic mother. There's some risks to this, obviously, because it's not always successful. It can be really expensive, but also there can be multiple pregnancies and multiple births as well because of the number. There are two types of reproduction: asexual and sexual reproduction. And if you're a triple scientist, you need to know the advantages and disadvantages of both. But if you're a higher combined or a foundation student, you just need to know the differences between the two. So the key way to know the difference is to look at this cell division processes, mitosis and meiosis. Mitosis is a great example of asexual reproduction in cells. And this is when a cell will divide to produce identical cloned copies of itself through one division, and these will form diploid cells. However, in the case of meiosis, this is when gametes are formed, and this is for sexual reproduction. So those gametes, that sperm and egg cell in animals, or pollen and ova in the plants, for example, these are a way for variation to occur, because each of the cells that are produced, the 4 daughter cells as opposed to 2 in mitosis, the 4 daughter cells that are produced in meiosis are all varied. And this is because the crossing over process of chromosomes to increase that level of mixing of the DNA. So it's all slightly different. These cells are also haploid, so they only have half the genetic information. So in the case of humans, a haploid cell has 23 chromosomes, whereas in a diploid cell it has 46 chromosomes, or 23 pairs. However, remember, this can be vastly different in other organisms, animals and plants, etc. So just be aware that haploid means half and diploid means full amount. Now, some of the advantages and disadvantages of asexual and sexual reproduction. So sexual reproduction leads to variation. So the animals or plants or organisms are able to adapt, and therefore the risk of disease is low. So one disease will not kill off all the species, or all the animals, or all the plants, or all the individuals in that species. However, disadvantages: it takes time and energy for an individual to find a mate. And if they are an isolated individual, they possibly won't find one, so they cannot pass on their genes and reproduce. However, in the case of asexual reproduction, population increase if the environment is good. So if it's a strong environment, the population will increase largely because every individual has the same genes, because they're essentially clones of one another. However, as there's no variation, this can lead to a disaster in terms of a disease that can wipe out the entire population. But again, there's no need for mates. Those chromosomes are tightly packed parts of DNA that contain genes that are small sections of that DNA. Now, if you are a triple scientist, you'll need to know that DNA is actually made up in the shape of a double helix, but it also is made up of 3 components, which we call together a nucleotide. A nucleotide has a deoxyribose or a pentose sugar. It has a base, which there are 4 different versions of. We have A, C, T, and G. A and T combine together or bond together, and C and G bond together. Phosphate groups are also the backbone of that double helix. So those 3 components make up that nucleotide. Now, the repeating unit of nucleotides help the genes and the DNA structure form the way it does. And we can actually take small copies of that DNA to synthesize proteins. And this occurs in ribosomes, which is found in the cytoplasm. So the DNA itself is copied into what we call an mRNA molecule. And this leaves the nucleus, where the original DNA is kept in the nucleus. The mRNA moves to the ribosome, and at that time, amino acids are produced as a result of each codon, which are 3 base pairs that equal 1 amino acid. And there's around 20 naturally occurring ones. And a string of amino acids, which are all combined together and chemically bonded, form a polypeptide or a protein. And depending on that sequence of amino acids will depend on the structure of that protein and therefore its function. So, for example, proteins include things like hormones. They control things to do with the body in the way of enzymes, hemoglobin. Gregor Mendel was the father of genetics. He came up with this idea that genes are the unit of heredity, that we can pass on our information through something called a monohybrid cross. Now, there are loads of different pedigree analysis, genetic diagrams that you need to know. Specifically, the 2 are family trees and Punnett squares. And depending on the level of GCSE you're studying, if you're a higher combined triple scientist, you may be expected to draw a Punnett square. If you're a foundation student, you just need to fill it in and interpret it as well. Some of the key terms that come up a lot and confuse students, we're going to go through. So genome is all the genetics of an individual of a species that makes it unique. A gamete is a sex cell, the sperm, egg, the pollen, the ovule if it's a flower or plant. The phenotype is the physical characteristics an organism has, and a Punnett square is just a genetic diagram. Heterozygous is 1 dominant and 1 recessive allele. Sometimes this is known as a carrier. And homozygous is when there are 2 alleles present, either both dominant or both recessive. So we have homozygous dominant, homozygous recessive. And we represent dominant with a capital letter and recessive with a lowercase letter. For a recessive allele to be expressed, we need 2 alleles to be inherited, whereas a dominant allele simply only needs 1. An allele itself is just a version of a gene that's inherited from the parent. So, for example, eye color, there are different versions of eye color: blue, brown, hazel, etc. And these are all the different versions. So they are all alleles. Now, in the case of this individual, we have two individual parents. We've got their genome or their genotype present here, and we have now filled in the Punnett square to show the possible offspring alleles. And as you can see, in the case of this Punnett square, we're crossing two plants. So we have a dominant allele, which is a white flower, a recessive allele, which produces a purple flower. As you can see, 3 out of 4 will have the dominant trait. And essentially that means that most of those individuals will have that dominant phenotype, whereas a recessive phenotype, as you can see, 1 out of 4, majority of those individuals will not have that purple color. So the recessive allele in this example, you can see, is less likely to be inherited because 2 alleles are needed. Now, in the case of family trees, you could be expected to interpret them and look at different family trees and predict who is more likely to be a carrier, who is likely to have the condition, and in what generation. So you can see generation 1 all the way to generation 4. Polydactyly is a dominant inherited condition, and essentially this causes an individual to have an extra digit or multiple extra digits on their fingers and toes. A recessive allele condition is cystic fibrosis, and this affects cell membranes, not just of the lungs, but also the digestive system. And treatments are needed for this individual to help them clear their airways, because infections can be quite frequent if not, as a thick mucus can build up and cause the airways to be blocked. Now, to test for this as a young child, if they're born, we can use something called a heel prick test, which is a small blood test that is done to check the health of someone. But also during IVF processes, this is often done as a screening tool to check the health of the individual. Additionally, you need to know about sex determination. So we know that XX chromosomes indicate the person is female, and XY chromosomes indicate the person is male on a genetic level. And it's important to know that there is a 50-50 chance of having a child that is male and a 50-50 chance of having a child that's female. And this occurs at every single fertilization event. So if someone's had a boy, they are still 50% likely to have a boy again. Just because they've had a boy doesn't mean they're going to have a girl next. It's always 50-50. Selective breeding is a process that humans have used to ensure that we have the best types of animals for farming, the best types of vegetables and fruits and other plants for agriculture as well, and also the types of domestic dogs that we see as pets in the home. And this is done through a process of, like I say, selective breeding. So we choose 2 individuals, a male and a female, who have the desired characteristics that we want. And then we breed them together to form offspring. From the offspring, again, those characteristics are selected and bred together. Over generations, this occurs to increase that trait so that it is more dominant in the gene pool. And over time, selection pressures mean that only those organisms with that particular trait survive and reproduce. And therefore, we have the desired outcome that we want. And this is particularly important for things like domesticated dogs and farming of animals. It's also useful in crops as well to ensure that the crops with the largest yield or the best ability to withstand certain weathering conditions are in high amounts. Now, another type of process is called genetic engineering, where essentially an enzyme is used to cut out a gene of interest, and the gene is placed into a bacterial plasmid, which is an extra loop of DNA found in bacteria. And because bacteria actually have the ability to divide rapidly over multiple generations through something called binary fission, not mitosis, this causes the increase in that gene being produced. Now, once that gene is produced in high amounts, it's actually inserted into an embryo at the early stages of development to help that individual produce their own version of that gene and incorporate it into their genome. So this was an example of how insulin has been utilized to help people with diabetes. We also have the golden rice product, which is genetically modified to increase the level of beta carotene. The first theory you probably know about is natural selection. And this was the idea by Charles Darwin when he went on his voyage around the world, specifically to the Galapagos Islands on HMS Beagle, the ship, in the 19th century. And he wrote up his ideas 28 years later in a book called Origins of Species in 1859. And he decided that evolution, all living things, essentially evolved over time because individual species have variation amongst their individuals in their population, and the best adapted will survive, breed, and pass on their genes to the next generation, meaning that those dominant traits become more increased in the gene pool. Other theories and scientists related to this include Lamarck, who had the idea that characteristics mostly used by organisms become dominant and passed on to offspring. However, Wallace kind of more agreed with Charles Darwin. He had this idea of speciation, and they both actually collaborated on this idea of natural selection. So what is speciation? If you're a triple scientist, you need to know about this. So speciation is when a species of organisms can actually breed together and produce fertile offspring. So a new species will form when a group is geographically isolated. So you have a population with a lot of variation, and there are a split and divide between that population. And this is because of either literally a land change, some sort of environmental change between the two populations. It causes them to divide. And due to mutations, this actually encourages environmental beneficial genes to thrive. And as a result, certain alleles will help those to survive more in those specific environments. And therefore, we now have two populations that actually can't anymore interbreed. And this happened in the Galapagos Islands. We had lots of birds, specifically finches, that Charles Darwin identified, having the same kind of origin. However, over time, they were developing their own specific beak shape based on the food availability. And that formed different species amongst them. Now, evidence of evolution, everyone needs to know this. You have fossils, you have bone records, you have tools, and geologists and archaeologists actually can identify the age of these specimens based on how lower down in the Earth's crust they are. However, fossil records are incomplete, so we're constantly finding new information to update our understanding. And we can also use antibiotic resistance as another example to show that evolution occurs through mutations, as all individuals of one species will die out, such as the dodo, a common bird found on the island of Mauritius. When humans and dogs arrived on the island, dogs were the new predator that wiped them out because they were a flightless bird. It's the same with dinosaurs, with the potential theory of the asteroid, and woolly mammoths with the ice age as well. There are lots of theories around extinction, and we're not quite sure what happened in lots of situations, but we can use data and evidence to help us, again, with fossil records. Now, in terms of extinction, not only is it a new species, it could also be a case of a climate change, like I said in the case of the woolly mammoth, but also competitors that can actually outcompete the ones that are best adapted. And this links to natural selection. Now, scientists have actually developed a way of categorizing organisms based on their specifics and the way they are, the phenotypes. And we do this in what we call the Linnaeus classification system. We actually do have technology now that uses genotypes, the genetics of that individual, to find out the origins of them. But back in the day, we didn't have that technology. So we use Linnaeus' classification system. And we also use Carl Woeser's idea of domain. So let's talk about this now. Now, you need to know the order of this. So domain, kingdom, phylum, class, order, family, genus, species, with genus and species being used to write the binomial name of an organism. And you can see the different categories will help to categorize individual species. Biotic factors are all the living components of an ecosystem. So the prey, the predators, the producers, the territory, and the mates. Abiotic factors like shelter, water, mineral ions in the soil, warmth, and sunlight energy are all the non-living parts that help impact upon the living structures within that ecosystem. Now, based on the environment will determine the adaptations that those organisms have. An adaptation, remember, is a change to an organism's phenotype based on the genotype, and is often driven by evolution and mutations. So mutations can be beneficial and help those adaptations to increase in prevalence. So, for example, desert animals and plants will store lots of water, in the case of fat, if it's a camel, in the case of tubers underground, if it's a cactus. Long eyelashes, again, to prevent sand getting into the eyes, and wide feet to stop the animals sinking into the sand. They also, if there's a plant, has spikes to avoid being eaten by herbivores. In the case of the opposite, the cold temperatures, we need to know such as the winter, polar bears have white fur to help them stay warm, but also to camouflage from their prey. They also have black skin to absorb heat to stay warm as well. Also, we have extremophiles, and these are often bacteria that live in incredibly extreme conditions and have adapted to survive in them. Now, food chains and food webs are ways for scientists to see how different organisms interact in the term of how they interact in terms of feeding, the relationships they have. So food chains and food webs always start off with a producer. So this is a green plant or algae, and this can photosynthesize and harness the sun's light energy. And this is eaten or consumed by a primary consumer, which is a herbivore. And this is often the prey for a secondary consumer, which is often an omnivore. This can also be a predator because it's attacking the prey, the primary consumer, but it's also the prey for the tertiary consumer or the apex predator, which is often a carnivore. And like I said, a food web just shows the interaction between multiple food chains and how removing one of those organisms can actually have a detrimental impact on all the organisms as well. So it's really important to understand those relationships. Now, there is a required practical here in terms of sampling, so using quadrats to measure the abundance of an organism that's often slow moving or not moving at all, really. So plants are a great example. Quadrats are just a square frame that help to find the proportional mean abundance of those individual species. We also have a transect line, which is used to measure distribution, and this is often used to see how, for example, on a sandy beach, how plants have changed in their distribution along that beach. Essentially, a transect line is just a measuring tape, and the quadrat is moved along it at regular intervals. And what we do is we record this information. You should be expected to know how to calculate the mean for both of these. The trophic level just tells us each individual level of a food chain. So trophic level 1 shows us the producer, trophic level 2 shows us the primary consumer, trophic level 3 shows us the secondary consumer, trophic level 4 shows us the tertiary consumer, and so on and so forth. Now, involved in this is decomposers. We'll talk about those later, but they essentially help to break down plant and animal tissue. And the living tissue that's recently alive or is living currently is what we call biomass. And we can actually plot this graphically in what we call a pyramid of biomass. And the length of each bar is actually proportional to the amount of biomass available at each trophic level. So you need to be very accurate when you do this. And you'll be given like a scale, or if you're a triple scientist doing the higher paper, you'll need to draw that scale potentially as well. Make sure your bars in terms of height are all the same. You've labeled it. Those are really key points to mention. Notice that as you go up, it is a pyramid shape because only 90% or 10% even of that biomass is actually transferred to each trophic level. 90% is lost through urine and feces, water, bones, shells, and other hard components, and carbon dioxide during respiration. So it's a very inefficient process, and that's why the producer has to be eaten in high quantities compared to, say, the primary consumer. Another key component of this that everyone should know is the carbon and water cycles. Carbon cycles is just how carbon is cycled throughout an ecosystem, and the same for the water cycle. There's lots of different components here, but just be aware photosynthesis absorbs carbon dioxide and respiration releases carbon dioxide. Carbon dioxide can also be released into the atmosphere when we use fuels and burn them through combustion. And fossil fuels, remember, come from dead organisms from millions of years ago. They contain carbon when we burn them because all organisms contain carbon on Earth. And decay, again, will release carbon as they will form, the fossil fuels as well. Now, the water cycle, there's a few different components here. Remember, condensation formed from water vapor cooling down and forming clouds. It can be transported across the world or different areas, transporting those clouds, and then rainfall in the form of precipitation. It can lead to infiltration, so water is stored underground in aquifers. We also have surface runoff, which can be absorbed by plants. Plants absorb water through the roots and release water vapor through transpiration stream. And this can also, be condensed again, and it cycles around. So humans can impact the environment around them. We know this because population growth has increased. That means more farming, more pollution, less resources, and therefore climate change as a result. Remember, there are lots of different types of pollution. So we have water pollution, which can lead to eutrophication. This is a process where fertilizers and sewage leak into rivers and water sources and cause an increase in nutrient growth for algae to form. Algal bloom will occur on the surface layer of that water and block out any of the light for lower species that are underneath in the water. So any of those plants and other organisms that require that photosynthesis from the sun. And as a result, those plants will die. That means there's less oxygen in the water, so fish will die and other aquatic animals as well. So this is really detrimental. We also have air pollution. So the combustion of fossil fuels will lead to things like carbon soot. If it's incomplete combustion, this will lead to things like smog and asthma and other health respiratory conditions. We also have carbon dioxide that's released in the case of combustion if it is complete combustion, and this can lead to global warming as carbon dioxide is a greenhouse gas. It traps that heat energy from the sun and warms the climate. We also have sulfur and nitrogen oxide, both of which will cause acid rain, which can cause fish to die and change the pH of lakes and rivers, and also destroy buildings as well and kill trees. We also have land pollution, so landfills where lots of the rubbish will go. If we don't recycle it, it can actually release methane, which again is a greenhouse gas causing global warming and that greenhouse effect. Now, deforestation links to this because trees naturally take up that carbon dioxide. However, if we cut down trees, deforestation for farming or space and resources, it can also lead to global warming, which essentially is the rising temperature of our planet, which can lead to drought, ice caps melting, and destruction of habitats. Now, another problem with this is something called peat bogs. Now, these are naturally occurring swamp-like structures which have very low pHs. They're very acidic. They have less nutrients there. They store carbon. They're essentially what we call a carbon sink, and this causes decay to slow down. And this is essentially a great opportunity for people to take some of that peat and put it onto their garden and encourage the growth of organisms. However, because people have done this over time, peat bogs have actually been destructed, and this means there is less biodiversity. They've also used this as a fuel. So peat has been used as a fuel because it contains a rich source of carbon, which is great as a fuel. However, it releases greenhouse gases, so it's not ideal and doesn't help the environment. Now, how do we maintain biodiversity? We obviously want to encourage growth of trees and less deforestation, encourage growth of plants such as hedgerows. We also have laws to protect endangered species, breeding programs for endangered species, seed banks for rare species of plants as well, and also recycling can help. And that's it from me. I've been the GCSE science teacher and you have been curious. If you did enjoy this video, give it a big thumbs up, let me know, share it with someone else so they don't miss out on key revision, and please do subscribe so you don't miss out on key revision too. Thank you so much for your time. Good luck for your exams. Catch you in the next one.